Hi, Hung. You made it.
Hi, Wendy.
Yeah, I was a little bit afraid that you wouldn't make it today because I thought you were the only one not responding yet.
Oh, I saw it on my calendar, so I just do it. Was it supposed to be 28th instead of 21st? Was that the thing?
Yeah, so I will be in Geneva next week at the World Health Organization.
Yeah.
So I thought either somebody takes over the chairing or we move it 1 week.
Yeah. Okay. Sounds good.
You need to take notes.
Yep.
Okay.
Got it.
I copy pasted already the things from last week into the Zoom meeting details. So in the monthly Mycetoma meeting from 21st of March. So I'll just wait 1 or 2 minutes to see when Matt comes in and Dimitri. They said they would be in, so.
Hello, hi everyone.
Hello.
Hi.
Hello.
Okay, then I think most of us are here. So let's start for today. And welcome to everybody, and nice that you could join 1 week earlier than scheduled. And I'm sorry for the inconvenience. I will be at a WHO next week for an integration on skin NTDs. So today we have several issues on the agenda. And the first one is the Fenrimol paper. And the first action item was for Dimitri and Hang to complete the SAR gap analysis. Could you, Hang, would you be able to give an update on that?
Sorry, I thought this item is actually done because the gap analysis is already done. So I think it's up to the stage where someone just need to send this, but I don't think it was tasked to anybody.
So we need to make sure that the things we're comparing them to, right, are active compounds. The discussion we had last time was that if we're going to request a compound, that the compound that we're comparing it to is active in each case, and that therefore it's worth requesting it.
Yeah, so that would be from I'll share this one. So right now there's 14 compounds that were mapped to active compounds. Sorry, 1 second. So it was back to this one here that I shared on GitHub about 3 weeks ago. So we've got all of this one here, which is 14. So all of this plus D is 14 compounds, and all of these were mapped to 1, 2, 3, 4, 5, 5 active compounds. And then since we're only asking about 10, around about 10, so this is already 14. So, and then if we have some more compounds on A and C as well, that would make 17. So there's a total of 17 compounds in the library that we can ask. So yeah, that's the progress. But
so on this diagram, for each of the ones that where we're comparing it to a known compound, you've got the potency listed there.
So this one here, there is potency. Okay, there was not potency added to these 5 ones just yet. Okay, so yeah.
Okay, so just so long as we're clear that all the ones where we're saying, well, we've got this compound and we want this compound, so we've got X, right? And what we want is compound Y to fill in a gap. We just need to be 100% confident that X, we know what the potency is and that it's to some extent active.
Yes, so these are very active. So these have all made it to in vivo. And 2 of these are the initial lead, the initial, like the very first lead as well. So
okay.
Do you want me to add like the actual numbers in? Is that? Yeah.
So just to make sure that all the compounds that we're using in comparators and we're not requesting are active, and we're sure that the potency is there. So just put the potency there, and then we're absolutely sure. And then we're going to be picking a small number of the comparators, which will expand it where it's worth going for, and then we can finalize the list.
Yeah. Do you want me to narrow that to 10 exactly? Because we have, I would say 14 to 17 right now.
Yeah, that would be good. If you put the top 10, then we can think about it. And then just dump that diagram in the issue for today's meeting, so we all know, we can all find it in the right place.
Yep.
Cool.
Okay, done. I'll test that to me.
And who's the one when the list is final to get it to ask Epicam?
With the smiles list, I can ask that, no problem.
Okay, so then we could put that as an action item for next time.
Okay.
Then the next query on the Fenrimol paper was if the chemical section was complete.
Okay, I met with Dimitri last week, and we have talked about it. We've cleared the majority of the comments. I have already made an update. I think it was, I haven't checked with Dimitri yet, but I think there was still
significant figures are left, and then we're clear.
Yeah, so there
Where
are
is this paper being hosted?
So the papers is, I don't have the link right here, but it's posted in one of the GitHub issues.
Oh, okay.
So I've already made my updates to clear some of the comments. I think it was now on to Dimitri to do the significant figures. So we've agreed that we'll make everything 2 sig figs. So I've already made mine to 2 sig figs for all of them. So it's Dimitri's now to also update his as well.
And we combined during the meeting, we combined all the methods together now. So there's no mismatch between UCL and Sydney methods as well. They're like unified now.
Yeah, and there are only two unresolved comments left, but those are for references. So we've already agreed last meeting that we'll do that at the end. So apart from the SIGFIX one and the 2 references comments, all of the other ones are done. So it should be ready for review once Dimitri updates his as well.
I will post
Okay.
the readiness of the paper like on GitHub as soon as I double check the significant figures.
Okay. And then there was an issue on the supplemental files of the manuscript. So the lab archives offline notebooks are needed. So both Dimitri and Hang know how to do this, I assume.
Yes, I've already I already uploaded my offline notebook to the shared folder for our manuscript. So it's now up there. Should have my name and lab archives on it.
Okay. Dimitri, and you?
Are we talking about the NMRs, supplementary information or something else?
The lab archives offline notebooks.
No, I haven't updated this yet because I'm still not sure like how to. So is it like PDF version or what?
No, it should not be a PDF version. It has to be an offline notebook.
Okay.
Yeah, so when the next version of the manuscript is there, I will go through the manuscript to double check all the molecules. And then I will make an offline version as well, an Excel on the biological data. So I will take that from the spreadsheet currently in GitHub and download it, make an offline Excel to fix that with the manuscript as well as a supplemental data.
Yeah, I can quickly, if I can just share the screen quickly, I can quickly show Dimitri how to get that.
I can see that. I can see that because I'm currently in Dropbox.
Okay,
I can
okay.
see the file. Yeah, I
No,
just
as in
found
like,
it. Yep.
do you know where to get the offline notebook? Because I can just quickly share it if you're having trouble finding where the button is.
So that's from Lab Archives, right?
Yes.
I think it should be fine because
Okay.
I did it previously. Yeah.
Okay, no worries.
Then we have to, well, when I'm preparing the Excel sheet, at the same time, I will double check that all the Fennery moles made are tested in vitro and in vivo. As far as I know, Ma, we tested them all. Is that
We
correct?
still some still 10 compounds from Fennery moles will be tested in vivo this summer, and the others, some MIC50 data needed to be added, and all experiments are going on. That's it.
So yeah, the MIC50 data, probably that will not take too long.
Yeah.
Do we want to include the in vivo data as well? Because that will take another summer. Matt, what did we agree upon last time? That we take a cutoff now or that we wait for another round of vivo experiments?
For the Fennery moles, geez, what kind of time are you thinking? Would that be like July or something?
No, that would be till the end of the season, probably.
Because I think we've already said last year that the cutoff was the end of season last year. That's all.
Yes, I thought so as well.
I think that's better, yeah. And if of
otherwise,
course, if we're asked for changes to the paper and it's delayed in review, then of course we could add more data in, but otherwise we go ahead.
Yeah, because I think otherwise it will just keep going on to next year and then it happens again. So
yes, I guess so.
Make it a point to cut it and then we can add something later for revision or something like that.
Yeah, the only thing for the extra compounds of the SAR, we do that in vitro then only and not in vivo, I assume.
Yeah, for sure. If something amazing comes out, we can test it later, but for the paper, it's in vitro.
Okay, yeah. Okay, Matt, so that saves you some time. You can focus for now on the vitro. And then the last action item of the Fennery Mall was, if all the above were done, then Matt is going to review the paper one more time. And probably that's also the time when the references either before or after are fixed. And also, because it's going to a chemical journal, probably the molecules are then renamed 1, 2, 3, etc. Okay, then that's on the Fennery moles. Is there somebody else who, is there something else we need to discuss on the Fennery moles for now?
I think we're
If
clear
not,
on that.
Yeah, if not, then we could go to series 2, the aminothiazoles. The first action item was that Rowan's contribution of compounds, the number of compounds, screening capacity, costs, and modes of interest to be discussed. Well, Rowan is going to ship some compounds to us. Not sure if those were these compounds, but it's in the process. I didn't receive them yet, but we were in contact. Matt, do you have any updates on this?
No, I'm not sure if it is series 2 or not. I can't remember. Do we actually have a sheet of the compounds he's sending, or is he just sending something? I'm not sure I've seen that.
No, he asked me if we would prefer loose compounds in glass vials, or if it's okay to plate them in a 96 well plate. Well, of course, we are more than happy to receive a 96 well plate. And then he's going to add some extra compounds. So he was going to make a map of that and share that beforehand so that we know what we will receive.
I have a question. Are you sure these are series 2 compounds? Because I think I remember he wanted to send some phenothiazine derivatives.
Yeah, I copied this from the last meeting.
Oh, okay.
I'm not sure if they are aminothiazoles. It looks like he has a larger collection, so I think it's more a collection he's sharing.
I think it's like mostly natural products. I'm not exactly sure, but there was like a phenothiazine like natural product in the library. But yeah, I think it was mostly natural stuff. I'm not sure.
Yeah.
So then we need to rename this one as under another series.
Yeah, from memory, I think it's like a collection of potentially a variety of stuff, but I don't remember which things are in there in particular.
Okay, I'm just sending an email to ask for the spreadsheet.
Yeah, he would share that before he shipped anyway. So
Okay,
then the true comment on series 2, I think that's for the different high schools. Did anyone have contact with them, or did Kimberly have, so that they all have their own targets to build or to make?
don't know if Kimberly had that meeting. That was the intention, but I don't know if that's taken place. She hasn't responded to this here. It would be good to know that. Hong, if you see her, would you be able to mention it and just say to what extent are the schools coordinated, and if they're not, maybe we should meet with them all.
Hung, did you get that?
I actually haven't spoken to Kimberly in a while. I can send her an email and ask about it, but yeah, because she's not in USIT anymore, so it's gonna be email correspondence for now.
So did someone take over this project from Kimberly at the University of Sydney?
Currently, I think Clem would be closest, but I don't think we have like a postdoc type of person taking responsibility for the project for this year. But I can double check with I should be about I will have to double check that with Peter and Alice just to make sure.
Yeah, because if somebody takes over this part of the work from University of Sydney, it might be good if he or she joins the meeting.
Yeah, I don't think we have like an official person taken over yet.
Okay.
Let me double check that with my supervisor.
Yeah. And it seemed the last comment on series 2 was that there would be a group of students at UCL, so in London, to work on this series. Is that correct, Matt?
Yeah, so it's looking likely that we'll get a few master's level students for a couple of months in the lab over summer making the immunothiazoles. That's currently looking promising. And I'll be able to give more clarity on that in a month or so, but yeah, it's looking good for that. In addition, Whitgift School have updated me to say that they've made a bunch of compounds and that we're going to try and arrange a visit for them to UCL so they can see, what we're doing. But they seem to be pretty happy with how the chemistry's gone, and they're gonna bring some samples with them, I think. And we're just trying to set up a date when that could happen.
Okay. So there will be some updates for series 2. And then if there's nothing more on series 2, we will proceed with series 5. Series 5, Ma, you tested some of the series 5 compounds against Falciformispora senegalensis.
Yes.
Do you have some initial results or something to tell everyone about it?
Yeah, just in the final, just 3 compounds inhibited both MM and FS. So yeah, it's a update.
So there are 3 very promising compounds. We did have more actives against Madurella, but in the end, of course, the ones which are active against both species are the most promising, because in the field you not only you don't always know what the causative agent will be, so then it would be good to have a compound active against all. I think we have screened them, but we didn't determine the IC50 yet against Falciformispora. Is that correct?
Yeah, we didn't.
Okay, yeah. So that's on our to-do list still. There will also going to be a search for new oxazoles. And Bernard, do you have an update on that?
No, not yet, because last time I already said that Andrea and Charlotte and their groups will prepare more new benzoxazole derivatives in the spring and early summer, and they will share their compounds then also to the Mycetoma or MycetOS project. That's the current state, I know.
Okay. And Hang, there were 2 honor students from Sydney looking into this series. Any updates on there?
Okay, I think there's one of them was having issue with enrollment, so I'm not sure what was happening there. I will have to check with Alice, see what's going on, because I haven't seen them in the lab lately, so I'm not quite sure what's going on. But yeah, otherwise they should be joining soon. Also, they're doing like a again their joint they're doing a joint honor, so it's like a half synthetic, half chemistry communication. So I think I'm not sure which part they are starting first as well. So that could also be why they are not in the lab. But let me just double check that with my supervisor this Friday, because I haven't seen them in the lab. So that's the thing.
Okay.
Gotta have I'll give an up. I'll see if I can get them to either join or like just say something.
Then series 6. Dimitri, do you have an update on the series 6?
Yeah, just a quick one. It's about the fact that I've sent 23 samples to Rotterdam.
Yes,
So those
and we
have
received
Yeah, those have been
received?
finalized. Some samples had to be taken from the list because there was a problem with NMR, and I'm currently dealing with this. And there's definitely going to be another batch, but I don't know yet the final number because they're still like target compounds, so the synthesis kind of keep on going for keroxymes. And I'm also doing a bit like methodology as well.
Okay.
Sorry, Wendy, just a quick word on the lab archives. I was just wondering what happened there for the paper. So carbon-13 data was missing. That's why it has not been uploaded. So the analysis have been done in advance. All the supporting information schemes have been uploaded, but the lab archives entries for compounds that are in the paper were missing carbon-13 files. So that's why they have not been uploaded, but it's not a problem because I have them like on my computer. So that should be a very quick update and then it's done.
Okay. Yeah.
Yeah, so I sorted this now.
Okay. So those were the items we had on the different series. Just before this meeting, there was an edit on the MMV open box, the global health priority box. We did get that box. Madhu, do you want to tell about the box?
Yeah, we tested them against the Madurella mycetomatis at high and low, 100 micromolar and 25 micromolar. Then now it is currently testing by the FS. Then we will get the data next week against the FS.
So we did screen the boxes. We still need to upload the screening results in the master file. We will upload those under the tab MMV boxes. So we will add it as soon as possible. So for those who are interested in those hits, then you can take a look.
Is there anything special about the box? Any kind of theme to the molecules, or is it just more molecules that are of interest that haven't been?
I think it was just it was some weird, like there were some weird structures in there, but I didn't look at it really. There were some unusual things.
Yeah, they were from companies which failed the target or something. And they made a small box out of it. And we thought, well, we screened all the other MMV boxes. Just let these also be screened so that we will have as much compounds as possible to choose from later on. Then we are already at the any other business. So University of Sydney, do you have any other business?
No, not really. Not for me.
London, University College London.
I have just a quick question with Ben Perry non-gone from the project. Who should I refer to about calculating log D values? Because we're using StarDrop like for consistency, and it's a professional software. So all the molecules, all the new entries in the MycetomaTis spreadsheet need to have log D values. Is there anyone I can refer to now? Does anyone know?
Yeah, there was somebody who took over from Ben. She couldn't help with the statistics, but I think she can help with the log D. So we could approach her. She's also part of MycetOS.
Okay. Because it's better to use the same software, like we kind of agreed this at the beginning, because for consistency, they use like different versions, different softwares have different parameters, and they might give like different results as well. It's better to be consistent with this if possible. But I don't have access to StarDrop, unfortunately. That's the main issue.
So we could ask her. So yeah, let's do that.
Yeah, I forget it's Louisa Cruz, I think, isn't it?
Yes, it's Louisa. Yeah, thank you.
All right, yeah, she's happy to do that.
I have to leave early, but it was good talking to you. So see you all
See you,
later.
Anthony.
Yep, thank you.
Last word on the mycetoma spreadsheet. So all the entries from the latest batch are already like in the mycetoma spreadsheet, including the isomers. So they will, so all the information is there.
Okay, yeah. Bernard, do you have any updates?
Yes, right now that Anthony is left. He recommended some compounds I made recently, a new niclosamide derivative and quinoline derivative, nitroxyline. I've prepared them and they are ready for sending to your lab. I plan to send or dispatch them the package this week, later this week on Friday, so that they can arrive at your lab in early next week.
Yeah, Mars is in. I will be out, but Mars in. So
Yes.
if you put in the package not only my name but also Ma, then somebody will give it to her.
Okay.
Yeah, and I will pay attention to the delivery. By the way, we found the 4 nature products worked well against both species. Species. So yeah, they are promising to do the in vivo test if they are no toxic for larvae.
And then Walid, do you have an update, or do you would like to say something?
Yeah, absolutely. Thank you. So I did have a few questions. During an exchange with yourself and Dr. Todd, a number of computational methods and approaches were suggested. Obviously, I'm still working on producing them, but I did have a few questions in regards to a few of the points. Mainly, I'd like to know of the points that Dr. Todd suggested, like how I can prioritize them. So in other words, like which are of most importance and like most dire need to get completed. And then I had some more specific questions about the first and second points, but maybe we can just start with that. And I can actually review them if need be.
So it's a little bit vague, the first point, the second point. Could you specify more what the question exactly is?
Yeah, absolutely. So Dr. Todd suggested a number of tasks in relation to doing some computational analysis on Madurella mycetoma. So first was to generate a phylogenetic tree of the fungal P450s. Second is to assess the biological targets of the series of compounds and to perform some simulations to evaluate binding free energies. The third is to find some known fungal proteins of interest in other pathogens to compare their sequences with those of Madurella. The fourth is to consider a more target-based approach, which I believe the first 3 kind of lend to. And then the fifth was to develop a good AlphaFold homology model for I believe for the P450. So yeah, I just wanted to know kind of maybe some guidance on how I can prioritize these, what criteria I can use to identify proteins that are most similar to the P450s and
yes, so Matt, I assume here you're looking more into the CYP51 gene, so the target of the azoles and also presumably the Fennery moles.
Yeah, right. So Walid, your background's bioinformatics,
Yes.
right? So
Correct.
the idea of the the CYP51 phylogenetic tree would be useful. So how similar this protein is in this organism to proteins of surrounding organisms, that would be kind of interesting to think about. So that if there were structures of some of those proteins in the literature, we could focus in on those rather than the ones that are more distant.
Yeah, so the current homology models are often based on Aspergillus because it's the only filamentous fungus which has a homology model or has a model, a structure, sorry. I could post on GitHub the enzyme, so the NCBI number. What is the one for
There's
Madurella?
actually an issue already existing on that which exists on GitHub. Here's the link. So hopefully you can see this. Yeah, so there is an issue on crystal structures of azoles, and it has some information as well.
Yeah.
So I think that might be helpful, and maybe if we have any extra information, we can post it here as well, just to have like everything together, I guess.
Yeah, absolutely, I can do that.
So the tree would allow us to visualize, you know, which are the most similar. That would be handy, so that we can see, you know, of the structures in the literature, what do we have and how close are they to the one that we're interested in. You know, again, obviously in the absence of an actual structure of the protein that we're interested in, what can we do to approximate to it?
Yes, and if you want to know which species are genetically closely related to Madurella mycetomatis, there are some papers from our group and from Siebren de Hoog, where they did classical phylogeny, so looking at barcoding genes. So that might help you a little bit where Madurella fits in the tree of life. So it's close to Chaetomium species, to Tilavia species, but the other causative agents are a little bit further away. So that those trees probably help you. I can also link those manuscripts in the comment under issue 68.
That would be really helpful. I appreciate it. So I take it then that the phylogenetic tree is probably of the highest priority out of those tasks that you suggested earlier, Dr. Todd.
Yeah, I think so. And then if you've got that, then of course you can use that to try and help you generate a good homology model that could combine with the AlphaFold prediction. So then we would have a structure which we could then use to try and rationalize some of the SAR.
I have a different question. Has there been any progress about kinase inhibitors? Because that would be also a good research field for dogging.
Mickey, you're in the meeting. Could you update us on that?
Yes, well, hello, good afternoon or morning or evening. So in regards to the kinase inhibitors, we're currently trying to get a program running that is able to identify the kinases that are present in Madurella mycetomatis. And once we have identified those, we plan on working further with them and see if we can find inhibitors to tackle them. So that's more of a bioinformatical approach. That's one thing. And the other thing, or the second like pipeline that we're looking into, is that Wendy is, I believe, looking into the kinase inhibitor library, which provides us with a set of compounds that should target kinases, and that we just screen those as well within the MycetOS project.
Yeah, so as an update to that, I was able to secure a little bit funding to buy a library, but I was trying to, or maybe somebody can help me with that, to see if the library we selected before is still the most appropriate library, or if there are better libraries currently available which we can screen.
Which was the library you got?
It's also one of the issues.
The KCGS library.
I have a small library of kinase inhibitors for our cancer research projects. I can provide, if you want, maybe 10, 12 compounds.
Yeah, that could already help. And the other library, I think, was 200 compounds or so. I will have to go through the issues to see what the last issue was on that. But
I think it's the kinase chemogenomic set, right? The KCGS site set, I think it is. Was originally built by North Carolina, is that right?
yes, that one.
Yeah, that's very good. I mean, that's the one you want. And it was available for a few $1,000s, I think.
Yes, so at that time I couldn't afford to buy it, but I got some small funding now, which would allow me to buy it. So yeah, that's the idea behind it.
Oh, that's great. So, I mean, any hits from that, we could follow up with the people at University of North Carolina, Chapel Hill, because they probably have structures around most of those structures. So that will be, any hits would be pretty useful.
Okay, yeah. I think, Walid, were you done with your update, or did you have more questions?
Nope, I think that's it for me. Thank you.
Okay, then I think everyone has updated the others. Are there still other things we need to discuss? Maybe a question still for Hang. For us this weekend, the time changes. When is your time going to change? Because I assume that the meeting in April will be a different time.
In April, Sydney should go back to like a winter time. So I think it's gonna be, I can't remember.
Yeah, I think there's a 2-hour difference between us and you, because we move the one way and you move the other way.
Yeah.
So if I recall correctly, it will be 2 hours earlier for you in the evening.
That should be fine. Yeah.
And for the rest, everyone I think will move the same way, because we're all on the northern hemisphere. So for us it doesn't change, but for you it probably does change.
Yeah, it's in the diary, 25th of April at noon again.
Yeah.
Have a look. I think it should be on here. Oh yeah, it's 2 hours early for me. That's fine.
That's doable?
Yep.
Okay.
It's gonna be 9 p.m. instead of 11 p.m. So it's actually good.
It's much better then. Okay, then that's also settled. And then I wish everyone a very nice evening, morning, afternoon. And see you all next time.
Thanks all.
See you.
Thank you.
Bye.
Can you stay for a second, please?
Yeah, no worries.
Bye, Wendy.
Yeah.
When you added the lab archives zip files, did you add them selectively or the entire?
So anything that you want, you copy it into a fresh notebook. You can make your own notebook that is owned by you, because your current one should be owned by Matt. If you make one that's you own yourself and you copy all
So
of the
how exactly did you do yours?
Okay, I can show you. Give me 1 sec. Okay. Okay, so I made a notebook. So it's actually I created a brand new notebook that's for the manuscript only. So it's this one here. And you just basically copy all of the pages that you want into here. So like all of these are what I wanted.
So that's how you make them selective.
Oh, you want to know like how that actually happened. I
No,
think.
no. So in order for it to be selective, like
Yeah.
zip file, you need to create a new notebook,
Yeah.
add the files, add the entries from the paper, and then
Yeah.
compress it like in a zip file and upload it to Dropbox. Okay.
Correct. So yeah, so all of these are entries that I want that is copied into this fresh notebook, and then you just go to utility and then just do that create offline notebook here.
Because I couldn't remember like what happened to it, and apparently the carbon 13 was done much later than the rest of the characterization. That's why it was incomplete and not uploaded. But it's not a big deal because it's done anyway.
Okay, that's good.
Okay, thanks a lot. Then I'll see you in April then.
Okay, no, right, see you.
Do you need any additional help with the SAR analysis?
That should be
Because
done.
we didn't discuss this on Sunday.
It should be okay. I forgot to put
Okay.
the potency data. That's why I was stuck there. But yeah, that should be fine. I'll put it.
Do you need help with this, or are you okay?
It should be okay. Yeah.
Okay, if something then let me know. Okay.
Okay. No worries. Thanks.
Okay, thanks.
Yeah.
And I'll see you in April then.